Ma'am, please start.
Yeah, a very good morning to one and all present here. Meet our today's expert, Dr. Sheikh Abdul Hussain, sir. He is currently a senior scientist in Dairy Technology Division at ICAR, National Dairy Research Institute, Karnal, Haryana. He obtained his bachelor degree in dairy technology from Osmania University, Hyderabad, and master's and doctoral degree in dairy technology from ICAR, NDRI, Karnal. Dr. Hussain is involved in teaching of several undergraduate and postgraduate courses of dairy technology. Dr. Hussain has been working in the area of functional fermented dairy foods supplemented with herbs, probiotics, and other nutraceuticals. Dr. Hussain got several awards and fellowship to his credit during his academic research career, including gold medal, JRF, SRF, merit awards, best paper, and poster awards. He has also been awarded with Associate Fellow of NATSI 2020. He has guided more than 10 PG students and published more than 50 research papers, reviews, book chapters, and technical or popular articles. Dr. Hussain developed 8 technologies and assistant 8 commercialization of 4 technologies. He has handled more around 20 in-house and externally funded projects in various capacities and has been known for his contribution as co-PI in the World Bank funded project, National Agriculture Higher Education Project, which made a significant contribution in the output or outcome of ICAR NDRI. On the behalf of organizing team SGIDT Patna and NATSI and NADCL Baramulla Jammu and Kashmir, I welcome you, sir, for the today's session on herb supplemented dairy products and their health benefits. Now over to you, sir.
Thank you, Sonam. Thank you for the nice introduction. So, shall I start the presentation?
Yes, sir.
Okay. So, good morning, everyone. So, today I will be glad to deliver a lecture on herbal supplemented dairy products and health benefits in the summer school organized by Basu and his associates, and its associates. So, as the topic suggests, first I would like to focus on the importance of herbs, and then I will focus on the dairy products which have been supplemented with herbs and their health benefits. So we all know that nowadays there is a great demand for functional foods, so which are the foods having some health benefit beyond basic nutrition. And according to a market survey, the functional food market is around $1,267 by 2027, and among the functional foods category, nowadays there is a demand for the functional foods added with probiotics or herbs. Herbs being natural remedies, they have gained a larger attention nowadays as functional ingredients. So their usage in food products or their usage as nutraceutical is growing, continually. So let us look at before going into the functional properties of herbs, let us look at the classification of herbs. Herbs have been classified, on several bases. There are, as per Paswan et al., they have been classified according to their uses. They have been classified according to their active constituents, and they have been classified according to their, seasonality of growth, etc. So when we look at the classification based on uses, they have been classified as medicinal herbs, culinary herbs, aromatic herbs, and ornamental herbs. So basically, medicinal herbs, aloe vera, garlic, cinnamon leaf, brahmi, ashwagandha, etc. There are several, medicinal herbs which have been also prescribed in Ayurveda for different, treatments. Whereas we also have culinary herbs which we use in day-to-day life, like basil, mint, oregano, etc., turmeric, saffron, etc. And there are aromatic herbs which give pleasant aromas. They are also used in culinary purposes or for some aroma purpose. And there are ornamental herbs like chicory, arrow, etc. And according to the active constituents present in the herbs, they again classified as aromatic, astringent herbs, bitter herbs, which contain phenolics, saponins, etc., and mucilaginous herbs and nutritive herbs. Whereas, according to their seasonality and lifespan, they have been classified as annuals, biennials, and perennials. Annuals, they complete their life cycle in one season within a year, whereas biennials they grow for more than one season in a year, whereas perennials occasionally they grow in two seasons, but they only bloom in the second season. So these are the different classifications of herbs based on their usage and lifespan, etc. So when we talk of herb may not be valuable as we say. So specific herbs have specific parts which contain active constituents. So while uh, extracting active constituents of those herbs, these specific parts only targeted. For example, basil, oregano, thyme, etc. They most of the active constituents are present. Whereas when we take cinnamon and cassia, bark part it contains maximum amount of active constituents. Similarly, when we talk about ginger and turmeric the root part contains the maximum active constituents. And the bioactive functions of selected spices and herbs, like if you can take cinnamon, it is having potent antioxidant activity, and it all regulates level, etc. Whereas if we take garlic and ginger, they help in combating sickness and cold, and they also help in reducing cholesterol, LDL, and they also improve the heart health, and they also reduce the blood pressure, etc. Whereas when we talk about turmeric, it is very good antioxidant. Also, it is known for its effect in fighting Alzheimer's disease, as well as it is having anti-cancer. And herbs also contain antimicrobial properties. So they are again different herbs are again active against different kinds of microorganisms, which I have shown here. So based on these functional properties, herbs have been used in various dairy products to impart various health benefits. Like up till now, a few health benefits have been targeted by supplementing herbs in dairy products. For example, some herbs are added to provide or act as natural antioxidant. Some herbs are added to improve the shelf life, that is like they act as biopreservatives. Some herbs are added to dairy products to improve the sensory qualities, and some herbs, they have been added to impart some other like and immunomodulatory properties or some other some other functional properties. So there, there are herbal supplemented ice creams. There are herbal supplemented butter. There is herbal supplemented cheese, paneer, etc., available in the market. So when we talk about dairy products, herbs have been mostly used in ghee as well as cheeses. So you can see here different kinds of herbs and spices have been used in different cheeses. So okay, mostly cheddar cheese, which is highly sold, has been targeted. Later, after that cheddar cheese, there is Gouda cheese, etc., etc. They have been flooded with herbs in different ways to give different properties like antioxidant and shelf life extension, etc. And apart from cheeses, milk, as I told you, milk, yogurt, labna, lassi, srikhand, butter, ghee, ice cream, etc. They also have been added with the different herbs, and their functional properties have also been studied, maybe in vitro or in vivo models by different workers. So instead of talking, uh, talking about miscellaneous studies. So I thought of discussing about a specific study which I have conducted during my PhD, as well as I have continued during my career as a scientist. So I would like to talk about one herbal supplemented dairy product, that is lassi. So I have prepared aloe vera supplemented probiotic lassi. So I would like to discuss about this. Generally, when we discuss talk about supplementation of anything into dairy products, it looks very simple that okay, something has been added to dairy products, but then the thought process going beyond addition of some herbs into dairy products as any functional ingredient in dairy products, and the consequences which we have to face are the problems are uh, other things which we have to face will be different. So because it is not simply adding something into dairy products so that it can be fine, it can be easily palatable. While adding something to any food product, we need to look at, or we need to consider several constraints, like how it is going to affect the sensory properties, or how it is going to change these bioactive properties, and how easily it can be accepted by the consumers, or whether the consumers could be able to uh, accept it, or maybe it is having no acceptability at all. So those things we need to see. And when we add something to especially probiotic dairy products, there we should be more cautious. Like probiotics being healthy bacteria, they are sensitive to ingredients added to their matrix. Like when probiotics are present in lassi, if we add any herb etc., which is having antimicrobial activity, the growth of probiotics may be hampered. So these things we need to consider. So if we look at my plan, which I had taken during doing this preparation of aloe vera probiotic lassi, I have three concerns while preparing this one. The first concern is that when I add aloe vera to probiotics or probiotic lassi, the growth of probiotics may be affected. So when the growth is affected, their numbers will be decreased, or their functional property for which I am adding probiotics. Maybe I am adding probiotics to improve the immunomodulatory activity. So their functionality may be affected. So I may not be, meeting the legal standards if the numbers of probiotic bacteria is decreasing, and I may not be, giving the health benefit which I want to give to the consumers. That is, their immunomodulatory activity may be hampered if the probiotic growth is, altered by. Another concern is that dahi culture. So when dahi culture is present in probiotic lassi, or when I add aloe vera to this to this lassi having dahi culture, what may happen? This dahi culture may again hamper the growth of probiotics, or the dahi culture's growth may be hampered by the presence of aloe vera. Ultimately, probiotic growth may be affected, and again, it affects the functionality, and also it affects the sensory texture, other properties of the food products, that is lassi which I'm preparing. Another concern is that aloe vera as such cannot be added, because it may be having some microbial load or some other enzymes which may cause some changes to the food product. So it has to be blanched, or it has to be given sufficient heat treatment so that its undesirable effects will be minimized. So the best way is to heat treat it, but when we heat treat it, aloe vera, what happens? Its bioactivity may be hampered. Again, it will affect the functionality of the food product. The bioactivity means, again, I have added aloe vera to improve the immunomodulatory activity. So when I heat treat it, there may be chances that the bioactive components present in aloe vera may be maybe inactivated, and they may lead to a loss of functionality. So these are the three different concerns which I was having while preparing this product. So in order to address these three concerns, I have gone step by step. So that I would like to explain here. The first concern is that to isolate probiotic bacteria, which can grow very well in presence of aloe vera without any growth restrictions. For that one, I took some milk dairy product samples, and I have isolated probiotic bacteria from this one. So I got several colonies, and among those, I found that there are 23 isolates with gram positive and platelets negative. So I did the genus specific PCR for these isolates, and then the first concern I have addressed here for for these 23 isolates. So I have grown these 23 isolates in MRS broth as well as in skim milk supplemented with 20% aloe vera. Why 20% aloe vera? I thought maximum 20% aloe vera can be incorporated into lassi. Beyond that one, it will be having undesirable sensory characteristics. So I supplemented with 20% aloe vera in broth and skim milk. And I have grown these cultures by adding 1% and incubated for 8 hours, and then I saw how their growth is affected. So among these 23, I saw 5 cultures are having good growth characteristics in presence of aloe vera. They have more than 7 lakh CFU or around 8 lakh CFU. So I took these 5 cultures, and then for these 5 cultures, I did specific identification using API 50 CHL3. So I got the cultures like L. paracasi, L. plantarum, 3 species, and L. rhamnosus. And for these 5 cultures, I did probiotic attributes. So because whatever the culture I got, I must see that whether they are potent probiotic or not. So I did acid tolerance test, bile tolerance, antimicrobial activity, antibiotic susceptibility, cell surface hydrophobicity, bile salt hydrolysis activity, and then antioxidant assay. Also, safety attributes in DNA agar medium as well as blood agar medium were carried out. And I found that these 5 isolates are having very good probiotic properties, and they also save. Among these probiotics, I have chosen Lactobacillus paracasi because it is having better tolerance, acid and bile tolerance, bile salt hydrolysis activity when compared to the other. Now the second concern is that compatibility of these probiotic in dahi culture. Why? Because when two different cultures are present in the same medium, they may hamper the other's growth. One may hamper the other's growth. We need to see whether they grow in unison or they may be having any difficult differences to grow. So I have co-cultured. I took 3 different dahi cultures like NCDC 60, 86, 167, having these microorganisms, and I have studied, their compatibility in terms of agar-well diffusion assay, as well as I co-cultured them, and I saw whether this dahi culture hampering probiotic count or not. And I found that there is no inhibition zone in agar-well diffusion assay, as well as the L. KSA species, L. paracasi species is having its growth is not affected in presence of dahi culture NCDC 60, etc. So I took this NCDC 60 as dahi culture for like dahi, I mean lassi preparation. And the third concern uh, which I encountered was like heat treatment of aloe vera. So when I heat treat aloe vera, it may hamper the bioactivity. So before adding it to the product and before preparing product using aloe vera, I must see the kind of processing, that is heat treatment, which I am employing to aloe vera, having any effect on its immunomodulatory activity. So I have tested that using MTT assay. So I have heat treated aloe vera, like mostly for probiotic preparation. Generally, we sterilize the ingredients and then we add to the product. So I have sterilized aloe vera juice, and then also I have used different other treatments like saturation and mild higher treatment than saturation, like 70 degrees centigrade for 10 minutes, 80 degrees centigrade for 10 minutes, and sterilization. So I saw I have taken 2 different concentrations of aloe vera and did MTTSA. I determined the stimulation index, that is how it is affecting the immune cells, and I found that heat treatment has no significant effects on the bioactivity, especially immunomodulated activity of aloe vera. So these three concerns were addressed. So I got probiotic having very good growth characteristics in aloe vera, and Dahi culture, which is more compatible with the probiotic, and a specific treatment which is having no effect. I mean, no significant effect on the bioactivity of aloe vera. So after that, I went for product development. So this is the flow chart, and you all know. And I have optimized the probiotic, aloe vera supplemental probiotic lassi using RSM, response surface methodology of central composite rotatable design. So I took milk fat, milk solids, not fat, and aloe vera juice as the variables. And these are the 20 runs I got for three different variables. And I have conducted 20 trials and sensory parameters, physicochemical parameters, as well as probiotic count, etc. I have studied, and I saw how aloe vera or its aloe vera milk fat and milk solids not fat, and their combination are affecting all these characteristics like sensory characteristics or chemical characteristics or probiotic count. And so based on the results, I have set the limits like this one, and I mean for optimizing. So correct, all the sensory attributes were taken as maximized. Like we must have better sensory quality. And pH was taken as in range. Weighing up should be minimized, so it took minimized. And probiotic count should be maximum, so I took it as maximum. And so again, after preparing our product with the optimal values, I got these are the predicted values and this is the actual value. And after conducting t-test, the optimized solution which I found was milk fat 4%, MSF 10%, and aloe 16%. So with these three ingredients, we can prepare better quality, I mean, better acceptable aloe vera probiotic acid. So this is the approximate composition. After that, I went for validation of immune stimulatory activity in vivo. So because the ultimate study is to prepare a functional product which is having some health benefit beyond basic nutrition. So I did animal feeding trials. I took 90 mice divided into 5 different groups. So one is basal diet, another is having milk as a diet, another is having only lassi, another is having probiotic lassi, and the other fifth group is having aloe vera supplemented probiotic lassi. So after feeding for 7 days, they have been challenged with, 4 groups have been challenged with Shigella dysentery pathogen. And then I did dissection on second, fifth, and seventh day after challenging. So if you can see that the IgA concentration in here, AL2 is the level of probiotic less. You can see that aloe vera probiotic less in IgA is maximum in aloe vera probiotic less. Here there is some difference, but because after treatment, there will be variation in immunity level because the animal will be under stress, so there will be variation. But then ultimately, at the end of 7 days, you can see that aloe vera probiotic less is the highest IgA concentration when compared to the other groups. As well as I also studied the colonization of Shigella, how Shigella is colonizing in different organs in different groups. So you can see that in spleen, especially after 7th day, there is very less number of colonization in aloe vera supplemented probiotic lassi group, this group. And similarly, in liver, in small intestine, large intestine, also the colonization is lesser when compared to other groups. Like this Taylor study also how these parameters were changing quality parameters. During storage study, I found that even though the product is fine, it is having immunomodulatory activity, but there is some problem, like especially the storage life is very less. It is around 7 days or lesser than that one. So which is a deterrent for marketability. So preparing functional food is not only the thing, but then we must have a very we must have good shelf life so that we can market it for long distances or we can have better marketability so that better profitability. So again, I thought of working on this one. So I continued my work, and then I went for addressing the challenge of shelf life. So I thought of increasing the shelf life through different interventions. But before that, what is the cause of shorter shelf life that should be identified? So we identified that post acidification, which is very common in fermented milks, is a major challenge in uh, smaller, shorter shelf life of our product. And there is syneresis, there is oozing out of water from the casein micellar matrix because of increase in acidity during post acidification. So it gives a bad appearance to the consumers. Another thing is that there is higher growth of yeast and molds because in acidic environment they grow well. So this is one more concern. Because of these three things, the sensory quality is hampered. So people do not prefer to buy, do not prefer to taste it. And another concern is that probiotic viability when there is increasing acidity, etc. So the probiotic survivability will be decreased. So their minimum number cannot be maintained, which is again a marketability issue or legal issue. So I thought of addressing these concerns. So in order to address these concerns, I followed four different approaches. One approach is to like restricting the starter growth. I mean, the post acidification issue is mainly due to the growth of starter culture even at preparation temperature during the storage life. So we must decrease or we must hamper their growth so that acidity production will be lesser. So I thought of using copper as a preservative so that it will. I have seen in literature that when we add copper etc. to fermented milks, they specifically decrease the growth of hamper the growth of starter culture, especially lactic acid bacteria. So I took that as one intervention. And then to prevent yeast and mold growth, which is causing undesirable flavor effects. So I thought of using natamycin because it is having very antifungal activity. And in order to reduce the sedimentation or syneresis, I thought of using stabilizers. And then to improve the probiotic viability. Why? Because while uh, we have packaged our developed product in LDPE pouches, so LDPE pouches are having very good, air permeability or gas permeability. So oxygen, basically probiotic bacteria are, microaerophilic. So high amount of oxygen is again a problem for them for their growth. So I thought of using uh, oxygen tolerating probiotic bacteria, better oxygen tolerating probiotic bacteria, are using high barrier packaging material which can act as a gas barrier so that the probiotic bacteria will be surviving for long. So these are the four different approaches I have used. So this is how I modified my optimized product preparation. So I have taken stabilizers as well as copper and natamycin addition in packaging. Uh, and then after that, I went for a storage study to look how these approaches are, improving the shelf life of my product. Initially, I went for choosing probiotic bacteria with good relative bacterial growth ratio in the presence of oxygen. So luckily, the Lactobacillus paracassis strain, which we have isolated earlier, is having good relative bacterial growth ratio. So we thought of continuing with the same bacteria. And then to prevent the syneresis, we have used two different stabilizers like high methoxypectin and carboxymethyl cellulose. So we found that high methoxypectin at 0.3% level is better to reduce sedimentation and it is improving sensitivity score, especially flavor score, etc., or oral acceptability score. And so we have taken 0.3% HMP as one additive to improve the shelf life, and then I have added copper at three different levels, like two different levels, like 2.5 ppm and 5 ppm. And I found that copper addition is having some effect on especially this S dermophilis, which is used as which is used as starter culture. You can see that at 5 ppm level, the growth is lesser when compared to control. So we took 5 ppm copper addition as another additive for improving shelf life. And natamycin also used to reduce the eastern mold growth. So natamycin is again taken at 5 ppm, 8 ppm levels. You can see that tyrosine is a byproduct of proteolysis, especially these unwanted flora. When they grow, their enzymes will degrade the proteins present in milk, and then they release amino acids. Especially tyrosine is taken as a indicator. So tyrosine values are lesser in natamycin added samples when compared to control and sedimentation, etc. Well, because when there is decrease in, there is increase in proteolysis, there will be more oozing out of water or syneresis. So you can see that syneresis is higher in control, whereas it is lesser in 8 ppm natamycin added samples. So there is no effect on probiotics. That's a good thing, and there is no effect on Escherichia. But then you can see there is a significant effect on yeast and mold count. So when compared to control, it is having significantly lesser amount of yeast and mold count. That is the one which we want. So natamycin is having. So because of these things, the flavor score is also good in natamycin added samples. So we have taken natamycin addition in 8 ppm as another ingredient for improving shelf life. So after that, we went for choosing packaging material. So we took 4 different packaging materials. LDP, again, it is a control, and then we took PET, polyethylene terephthalate, and EVOH polymer and glass bottles. So we have these are the different barrier properties of these products. Then we have stored our samples in these materials by after adding all those ingredients like copper, pectin, as well as natamycin. You can see that the differences in pH, etc. So to surprisingly to our, the samples in glass bottles and EVOH, they are having lesser pH. So, so that is not a good indication. Uh, but then tyrosine value also higher in case of these samples, slightly higher compared to other things. We thought otherwise, but then we got new results. So, but interestingly, what we found is even though it is having lesser pH and higher tyrosine values, especially EVH in glass, the flavor score is higher in these samples. So the oral acceptability score is also higher. So it indicates that the kind of fermentation occurring in the presence of oxygen or in the absence of oxygen is different. So even though the pH is lesser, but there might have been generated the absence of oxygen might have generated some kind of substances which may be whose flavor is better than the LDP or PET based packaging conditions. So they have higher flavor score and higher oral acceptability score. And the probiotic count is also higher. Obviously, when there is less amount of oxygen being microaerophilic, they will be having better growth, better survivability. Again, eastern mold growth is restricted. So we took EVOH or glass, either of these as a better packaging material when compared to the PET and LEP. But then glass being easily breakable, etc., having issues with handling. So we thought of finalizing EVOH as a packaging material for enhancing the shelf life. So our shelf life, the product shelf life initially was 1 week. Now its shelf life has been extended, 24 days, 25 days almost. So that's a significant enhancing shelf life. So that is the study which we have done here at NDRI by supplementing carbs into daily products. So as a output of our research, we got some research papers and a review article and some book chapters. So thank you very much.
Participants, now session is open for discussion. If you have any query, you can ask to sir, or you can interact with sir. Or if you are not in good connection, you can put your question in the chat box also. Jannat ma'am? Yes, Kiso, you can put your question in the chat box. Hello? Please unmute. Jannat ma'am, please unmute the participants so they can questions. They can raise their questions. So there is question in chat box.
Yeah, I'm looking at it. The herbal medicine. The herbal medicine you suggested can be used in goats also. If so, please share it. Those and details. Goats. Okay. I'm sorry. This is not medicine. This is basically a functional dairy products which we have prepared. This is aloe vera probiotic lassi with extended shelf life. So regarding goats, and regarding medicine, so this is basically a food intended for human consumption. So I'm sorry. Aloe vera can be definitely used for different purposes. Not lassi, maybe for goats, for any goat treatment, वगैरह. So aloe vera has found its applications in animal health treatment also. So there are literature and there are prescriptions in Ayurveda. So you may look for that one.
Okay, any other questions? Participants?
I think some people are raising their hands, so you can unmute them so that you know.
Yes, we are trying so. Kishoji, can you ask your question in the chat box if possible, please?
Okay. Sorry, ma'am. I thought you were talking about the product. So you were talking about the initial slides which contain active principles of various. Yes, ma'am. Definitely. So herbs,
Participants, please ask questions.
yeah, herbs can be used for treatments for animals also, ma'am. Even for goats also they can be used.
Hello.
डॉक्टर शेख साहब
Yes, sir.
हेलो
Namaste.
या
sir.
नमस्ते सर सर
sir,
Yeah,
मेरा
please.
बहुत ही एक आसान सा एक सवाल है बेसिकली हम लोग रिसर्चर हैं हम लोगों ने वैल्यू एडिशन में हम लोग बहुत अच्छा काम कर रहे हैं आपने भी जैसा ये हर्बल में बहुत अच्छा काम आपका मैंने देखा है स्लाइड में भी लेकिन मेरा सवाल ये है कि हम लोग मार्केट शेयर हमारा क्यों नहीं आ रहा है